For these notes, you are going to need 2 pages. We're going to take a full spread, so we're going to be using both sides of our notebook. Our notes today are on differentiation. And differentiation is the process of development that turns stem cells into specialized cells. And these specialized cells can perform specific functions. So we're going to start off with a stem cell. And the stem cells are able to divide. So they are going to go through the process of mitosis in order to make more stem cells. So we're going to put a little note here, and I'm going to be switching colors so that my notes are in blue and my drawings are in this pink color. So stem cells are going to divide by mitosis. To create more stem cells. And it's important to note that all of the cells in your body have identical DNA or genes. So this cell can also divide because it's a stem cell, and you're going to get more stem cells. And every time it divides, it makes 2 identical cells. Identical because they have identical DNA. And these stem cells, they can divide indefinitely, which means forever, as many times as they want. Their job is to provide all of the cells that your body needs. So sometimes you are going to have cells that die. Cells just naturally have a lifespan. And if you have cells that are dying, you're going to need to replace them. If you get injured or damage your cells, you're going to need to replace them. So stem cells are going to divide to create those additional cells that your body needs. So this cell here is going to go through the process of differentiation. And once it goes through this process, once it actually starts to differentiate, it cannot go backwards. So as it becomes a specialized cell, it can no longer go back to being a stem cell. So differentiation occurs by turning genes on and off. So this cell is going to start differentiating. Some genes are going to turn on and some genes are going to turn off. And this process is going to result in the cell going down a path to become a specific type of cell. Now, if different genes had been turned on, then the cell would go down a different path. And once the cell starts to go down this path, it cannot reverse. So we're going to end up with, on this side, we're going to go down this path of becoming what's called a myeloid blast cell. If the cell had gone down this path, it would become a lymphoid blast cell. But we're going to go down this way, and this way is going to make our myeloid blast cells. Now, we know that blast cells are immature. They are partially differentiated. And what this means is that they are not fully specialized. And can still divide. And these are the cells that Hina has too many of. So if we're following the path for this lymphoid blast cell, then what we can end up with are actually multiple different types of white blood cells. So one type of white blood cell that's going to form, if the right genes get turned on and off, is called a natural killer cell. And natural killer cells, we're going to draw like this, because they have these chemicals in them that can destroy your body cells or other cells. Now, our other option is if different genes get turned on and off, you could end up with a T cell. And we're going to draw T cells like this. We're going to put these little receptors on them because T cells are responsible for identifying if there's something invading your body or if a cell does not belong. So those are going to be your T cells. And if even different genes get turned on and off, you might end up with a B cell. And a B cell is another type of white blood cell, like a T cell or a natural killer cell, but these ones have antibodies on them. And if they are activated, then another set of genes will turn on, and they will differentiate again to form another type of cell called a plasma cell. And plasma cells are going to actually release antibodies that are specific for the type of disease you've been infected with. So your immune system cells are going to work together and you're going to release these antibodies from plasma cells. Okay, now let's go this other way, and this is the way that we're going to actually follow. So I'm going to draw a solid line for some of these. So if I go through this solid line, that solid line is representing the genes that are being turned on and off to make our actual cell, and we're going to be making something called a megakaryocyte. So myeloid blast cells could, if they turn the right genes on and off, could end up developing into a white blood cell called a neutrophil. Or they could develop into a red blood cell. Or they could develop into something called a monocyte, another type of white blood cell. And this monocyte can develop into two different types of cell, depending on which genes are turned on and off. So this was a monocyte, and it could become a macrophage, one of my personal cell favorites. Macrophages basically eat things. They can chase things like bacteria and then eat them. Or if this other set of genes gets turned on and off, you might end up with a dendritic cell. And dendritic cells have these weird protrusions. Now I'm just kind of randomly making these. Hopefully it'll look okay. So this is my dendritic cell. And then the pathway that we are actually going to follow, so we're pretending that our cell actually turned on the genes to become a megakaryocyte. And megakaryocytes what they do is they release platelets. And platelets are just fragments of cells. And they're responsible for making it so that blood clots. There are actually a couple other types of white blood cell that can be made from the myeloid blast cell, but we don't have really room for them and we're not going to worry about them too much. So let's take a few more notes here. So first of all, I want to make a little note about the fact that, we'll do it right here, our fully differentiated cells so our megakaryocytes, our macrophages, our red blood cells, our T cells, these fully differentiated cells, they perform specific functions. Like your red blood cells are the cells that are going to carry oxygen around your body. Your megakaryocytes make platelets, which help your blood clot. Your T cells are a part of your immune response. All of these cells have specific functions. The thing is most can only divide a couple of times. And some can't divide at all. So your red blood cells, they can't divide at all. So if you need more red blood cells, you cannot have a red blood cell divide. Instead, you have to have a stem cell go through the process of differentiation to actually make more red blood cells. And that's the other thing. Differentiation, it cannot be reversed. Once the development has started, the cells are stuck on that path. So what this means is that if you need more red blood cells and you need more neutrophils and you need more macrophages, in order to make each of these cells, one stem cell has to go through the process of differentiating to make one neutrophil. And one stem cell has to go through differentiation in order to make one red blood cell. Now, it's possible that this myeloid blast cell right here, it could divide. Because it's a blast cell, it can still do division. And then it could make, from the two cells that it makes, one of them could become a red blood cell, one of them could go through this process of becoming a macrophage. But once the cell has started down this path, it cannot go backwards. This is a one-way street. It's like learning how to read. Once you learn how to read, you don't just unlearn how to read. Scientists are actually working on trying to figure out how to undifferentiate cells, because if we could go backwards in this process, then we could potentially turn developed cells into stem cells. And that would allow us to do things like if we needed to repair someone's nervous system, like if they were paralyzed, if we could get some nerve stem cells to go into that location and to differentiate, then we could potentially repair that damage. But right now, we haven't figured out how to do that. So once the cells are going along this path, they cannot go backwards. They will continue to differentiate until they become a fully specialized cell that has a specific job. Now, this process of making all these different types of blood is called hematopoiesis. And that is the differentiation of blood cells. Because you're an adult, well, because you're not an embryo, your stem cells are already partially developed. So this stem cell that we're starting with here is able to develop into all the different types of blood cell, but it's not able to develop into, say, skin cells or bone cells. You have different stem cells that are able to do those jobs. Embryonic stem cells, the stem cells in embryos, they can become all of the different types of stem cell. So that's it. Those are our notes on differentiation.